Hello, hello, and welcome to the Central Texas Mycology Society monthly meeting. We are going to be joined tonight by Ryan Manthei, and he's going to give us a little presentation, talk a little bit about cultivation and building the myco community here in Central Texas and all throughout Texas. So you want to take it away, Ryan?
All right. So howdy, my name is Ryan Manthei. I'm a cultivation specialist at Smallholds. And basically what I'd like to do is walk you through like some of the cultivation process in video, and then also kind of basically do a small lecture over some mushrooms, maybe do some microscopy if anyone wants to do that, and then finish with kind of how I make substrate to add the spawn to later. So what I'm probably going to use is chicken scratch. That's what I usually use. And give me just 1 second.
Compelling YouTube, everyone. Sorry, I guess he had to run and go grab something. Here he is. Something you don't want to keep inside.
So basically, this is just your regular Instapot. It's an Insignia brand. They're a little bit cheaper at Best Buy. They have a whole bunch of them overstocked, and they're trying to basically like give them away. I think this is like $30 there. I remember birdseed. You can mix it up with oats or something, but I usually just kind of do one whole pot. And basically, I mean, I fill it up to the fill line. Do not fill above here. And I pretty much just fill it up to just cover the wild bird seed. And I usually just run it for about 10 minutes. And that's usually enough to force enough water into the spawn. So now, while that just goes in the background, Well, that goes into the background. Basically, I'm just going to be going through a PowerPoint from one of my old professors, Dr. Rodriguez. He teaches the mycology course at Texas State. And I think he's really been kind of one of the main people that got everybody in this area into it. I know like Philip has experience with him. Pretty much everybody I've met has met somebody that he taught in a class. But basically, we're just going to go over Basidium mycota, mainly because that's most of the things that we eat are going to be from them, are going to be in the family of Basidium mycota. Those are the ones that have gills. It's the second largest group, very, very highly described. Most of the species we come into contact with are these ones. Although they are very, very diverse, they have all kinds of crazy different adaptations to persist, basically. And so basically, let me, so on the side over here, you can kind of see basidiospores and the basidium. Those are actually the spore-bearing surface on the gills. And so basically the gills produce this sexual reproductive structure that's different than the normal growing hyphae. And then that begins to differentiate out into the basidium, which then forms the basidia at the ends. And if anything interesting you want me to stop or explain it more, just type it out in chat. But basically, here's just the general life cycle of it. Here's some hyphae under a scanning electron. Here's it under a regular microscope. Then they produce the fruiting bodies. Then they produce the basidium and the basidiospores. And then eventually those spores land and germinate. One thing that's very, very unique that shows sexual mating is clamp connections. And so clamp connections occur whenever there are two different nuclei. So basically two spores have germinated, they have grown out hyphae, those two hyphae have interacted and touched each other, and they have transferred nucleuses, actual nucleuses. So then those begin to proliferate. Every cell after that will have the nucleus of both the negative type and the positive type. So if you ever put hyphae under a microscope and you do see this little nodulated bump with the double wall, that pretty much guarantees that you that mating has occurred for sure. And so a lot of basidia, a lot of fruiting mushroom will only fruit if they have mated. So basically, if you just have a block of hyphae and it won't fruit, one thing that's recommended to do is to take it and put it under the microscope to see if it has actually, if it does have two nuclei and can bear fruit, or if it's basically from a spore syringe. It's unlikely that one single spore would take it all over, but it is theoretically possible. And so there's a lot of different types of basidiospores. And so there's some that shoot out their spores. There's some that just kind of blow it away in the air. There's some that put it inside themselves. And so basically, if they are symmetrical, like these ones, they are forcibly shot off like a cannon. They're passively. If they're asymmetric, kind of like these ones over here, then they're usually shot off. And so there's a lot of different ways that they can actually be forcibly ejected. One is through a catapult mechanism that builds up dew pressure on the inside of the spore and eventually slingshots the water down and then forces the spore up with the equal and opposite reaction.
What do you mean by dew pressure?
So essentially the spores will collect water on the inside, and that will form a water droplet as more and more of it collects. And eventually that drop will become about the size of the spore. And when it does, there's essentially a very slick spot right at the joint hinge with the spore. And so once the water touches that slick spot, it shoots it basically down with the force of gravity. And once that mass of water is flowing down, it basically catapults the spore up and off of it. They basically swap momentum.
Cool.
Does that answer your question? Okay.
Yeah, that's very cool.
And so basically, I forgot the exact, okay, yeah. So basically, these are spores of each of these larger ones. So like, I believe this is like a puffball type mushroom. This is a stinkhorn. I think that is another puffball. That looks like it's just a truffle. And then some guild spores. And I think this little circle right here is supposed to be the dewdrop. And so basically, those were images of them up close. Oh yeah, here's the actual picture for what I was explaining. So along the gills, you'll have basically millions and millions of these spore structures that are the basidium. They almost always come with a set of 4 basidia at the end of them. And so essentially what will happen is the spore will build up a film, and then that will catapult and eject it off. There's a little bit more detail on it.
Hey, Ryan, someone was asking you to slow down a little bit.
Oh, okay.
Yeah.
Sorry, I missed that. So basically, there's this little segmented area that's the hilar appendage. And so that's kind of that joint I was talking about. Once the droplet builds up, basically whenever it touches the base, it basically flings the spore off. Yeah, and so this is basically a free body diagram of exactly how that slingshot mechanism kind of works. Although there are a lot of different mechanisms in Ballistiospori, and I believe some of these are, I know that a couple of these are mentioned. And here's some actual photo, like time-lapse photos of it happening. Here is it under a microscope. And so basically one of the type of basidia, the large structures that bear off the spores, this can really help you identify a lot of them. And so some of them are segmented, and some of them are asegmented or not segmented. And so if you're identifying a new species or you're seeing something funny under the microscope, that is one way to differentiate a lot of the different species. And as it's saying here, almost all of the basidiomycetes, including the agarics, which are most of the edibles, polypores, bullies, and puffballs, are without a septa. So it's just kind of one long body, or maybe not necessarily long, but it does not have different sections growing off of it. It's kind of a clump with all 4 extending off of it. And so here is basically some of the different ways that the spores relate to kind of their structures. So I believe this one is witch's butter. And so that's over here. It's very, very long. And this one usually forms when it's very, very humid, and is usually forming on the ground or on like woody structures. And so that's how you can see they are septated. So there's 4 separate nodules here that each go up and to make its own spore, instead of all 4 spores being connected to one plump undivided area. And so that is cruciate septate, basically up and down into different oval type objects, footballs almost. And then you have transverse septate, which is separating it longitudinally. And so this is a woodier, I believe. I don't know how to pronounce it, but the Aurucularis. And so essentially each one of these bears off a spore. And so those are the 4 different like septated areas, because I don't know why, but it's always 4 spores, which is kind of a interesting thing. And then tuning fork. So basically these 2 are coming off of a big plump area, and then they each have 2 off of them. So that's why it's called the tuning fork. And so it's almost always 2 sets of these together in like a double Y, but also called bifurcated. And I'm not sure, but I think this might be like a jelly mold, or a jelly fungi, or I don't think it's a slime mold. But yeah, and so they produce the tuning fork ones. So it's something I've seen all of these out in the wild. I don't think I've actually ever placed them under a microscope and tried to identify their different sporulating bodies. This is just kind of its subsection in the kingdom of fungi. And so this is the subkingdom Daiquiria, I believe. And Basidiomycota are the most recently diverged behind the Ascomycota. And so these are the newest evolved fungi in the kingdom. And within it is the Agaricus, most of the edibles, Utsinolog Mytina, which I believe is responsible for corn smut, and I forget what this one is, but it's another type of smut or rust. So okay, yeah, here we go. So that also, the Agarica Micatina also includes jelly fungi and yeasts. And Hymeneum covers a diverse amount of structures. This one is a smut fungi, and so I believe this might be the corn smut. It's considered a delicacy in some places. And then yeah, Puccino is the rusts. So some more of the things in Agarica Micatina, there's a lot of wood lovers, a lot of litter decomposers, and a small number of like molds and pathogens, some vegetable crop damage and some timber damage. Obviously polypores and wood decayers are not really what lumber seekers are looking for. There are a couple of human pathogens, although they're very, very rare. And you can actually do ink stains to find them. And some of them you can see under the microscope. And if you don't know, a telomorph and an anamorph are basically a sexually reproducing one and an asexually reproducing one. But neoformans are in the Agarica Micatina. And I've heard that it's very, very bad because it just spreads, and it spreads very, very easily and rapidly. So some of the Agarica Micatina are very poisonous. You can have like the Great Destroyer, the Death Cap, Deadly Gallerina. I believe this one's the Deadly Gallerina. Some can become poisonous over time. Some are hallucinogenic, obviously. And then some of them have certain things that will make them poisonous. I know like ink caps are non-lethal, but if you take ink caps and you drink alcohol, it can become lethal.
Okay, so that one, the purple one that you pointed to, that's actually the Lacaria.
Okay, thank you.
The one in the middle on that bottom row, that's the Gallerina, and the one to its left is the Psilocybe, and that's why you can tell they look alike, and that's why people recommend being very careful. And then
gotcha.
Yeah, the two deadly amanitas at the top.
The Death Cap and the Destroying Angel. Thank you.
Yeah, no problem.
And so this is some large cultivation of it. These are all portobellos, which I mean, they're known as the button mushroom. I think Cremini is separate from the portobello, but portobello gets sold as a lot of different things. You also have like the wood ear and shiitake. Both of those are commonly cultivated in Asia. The wood ears are highly, highly loved by them. I have a hard time. I'm trying to warm up to them because they are really, they're a very interesting texture, and they're full of a lot of immunomodulators. Actually, I think most fungi really are. So I'm just trying to diversify into all of them. But wood ears are pretty cool to cultivate. And then again, you have like the portobellos. And I believe these are basically just long runs of, usually it's either manure or some sort of like wood mulch compost. I know that Mike Tyson is doing several videos on it. His father, I believe, cultivated portobellos for about 50 years. And so he basically has some trials trying to do them with a bunch of different gourmet ones, and basically just a wide range of other mushrooms to see, to really try and get people away from bag culture and like monotub culture, and basically get off of our plastic and back into kind of a more traditional mushroom cultivation method. Yes, go ahead, Phil.
Oh, yeah, I was gonna ask. So they're trying that with like different species than even like the shiitakes, like oysters, or I don't know. Kind of.
Yes, I believe.
That's Mike Tyson, the boxer, right?
No, that's not Mike Tyson.
Oh, I thought. I was like,
sorry, I always say his name and I forget. No, MYC He basically, I believe he started
Oh,
Myco Bazaar on Reddit. And he's a very large proponent of mushrooms and just mushroom culture. He's really well known for lion's mane in particular. And basically just does a lot to facilitate kind of a community discussion about mushrooms. I do not actually know his first name now that I think about it.
word.
Well, I mean, I believe it's Mike. And I guess it's probably his last name is probably Tyson. And he just changes it for the business. Um, but anyway, he's a very, very awesome resource. He's a loving guy. Usually answers almost any kind of direct message on anything, and really just kind of out to help people. So here are some chanterelles and some boletes. I'm guessing this is the tricholoma. I know that there have been reports of boletes around the San Marcos Library. And I know chanterelles have been found this season around a couple apartment complexes in San Marcos. I don't remember the name of the apartment complexes, but we had a worker come in with some chanterelles a couple of days ago. I am not familiar with the tricholoma, but I would love to wild forage something like that or find something like that around here, or learn how to cultivate it. And so the Agarica family basically produces some of the largest fruiting bodies. And then not only that, I believe these are also the largest living organism is basically the honey mushroom. And that one is like estimated to be like, I don't remember the number, but several hectares, I think, of just one continuous single organism. So they're in my opinion, they're kind of they're definitely the most viewed family. And so here's a breakdown of kind of some of the different orders that are contained within Agarica mycetes. So you can see the boletes, the Agaricus, Rusulailies. I don't know how you say that. I always call them Russells in my head.
Rusolailies.
Thank you, Rusolailies. That sounds so much better than Russells. The polypores. I believe these are like the stinkhorns. This is the Texas star, I believe. Then the gasters. This one, I remembered it earlier and I already forgot it. But the chanterelles. So all of these are in the Agaricas or the Agarica mycetes. And that is the end of the lecture. There's also a second part. If anybody wants, I can go through that. Or I can go on to basically how I make substrate if y'all would like to see that.
I mean, I'm down. I love learning. I love seeing evolutionary trees.
Okay.
If people want to go into that, if I'm the only one speaking up, then I say we go for it.
Okay, here's where he's talking about the different evolutionary timelines. So jellies diverged earlier. And so some of these have different types of forms. They have both a jelly and a crustose form, I believe. And so some of them have double, and I'm not really sure how I track that back. But there's a lot that's not really understood in the evolutionary timelines of mushrooms. A lot of traits seem to kind of jump around. So it's almost, so what he was kind of hypothesizing is that they really retain a lot more control over their DNA than most organisms do. So even though a gene has been turned off and is no longer really kind of actively being shown in the phenotype, as long as it is still retained in the genotype, it can go back into its genetic memory essentially and turn on genes and begin to actively kind of select what direction it wants to go in, which is funny to me because kind of one of the original theories of how evolution worked was like they thought that like the giraffe just wanted to reach the leaves. And so that like by wanting it or by desiring it, that like increases chances of growing longer. Which I mean, we don't really know how the mind works, but we're pretty sure that's not how giraffes' necks work. But to see something like that in actual mushrooms where their desire can actually change their genome is pretty astounding to me.
There's also a lot of convergent evolution that goes on in fungi too, where like certain morphological structures will show up in different evolutionary lines just simply because like they work so well,
Mhm.
that like they get selected for again throughout the environment. That's another way to
Mhm.
think about that too. Is
Yeah.
like it's the strategies like for the jelly fungi, they work so well, so they show up in different aspects of the tree as like they're going through different diversifications and moving into different environments, but like it just seems to work. And then there's also evidence of like of kind of what you were just talking about, of like this gene retention where like they'll actually evolve into like a certain structure and then they'll evolve kind of back out of it.
Yeah.
uh,
Mhm.
so it's pretty interesting stuff, and like that's one thing that we're learning now as we like do more DNA analysis, is like learning that some of our morphological phylogenies, these trees that we're looking at, aren't actually what is the evolutionary reality, and that
Yeah.
tends to throw people off. That's where you get a bunch of name changes and stuff like that. But
Mhm. Well, and also just how we make clades is prone to a lot of rewriting, but it is like the best tool that we have. And also, I realize I anthropomorphize mushrooms a lot and say that they like desire changes. But yeah, basically that they have much greater control over reactivating those past genes. Um, but yes, here are some more of the kind of locations on the trees. So these are the oldest, or wait, uh, yeah, these are the oldest and these are the youngest. And so basically, you can kind of see at least a few things that stick out to me are a lot of the things like the gasters and things like that that really help with ectomycorrhizal stuff. Same thing with the chanterelles, or the decaying of very hard woody surfaces like the wood ears and stuff. To me, those show that mycology or mushrooms in general really kind of had a huge birthing, or at least the orders that we see had a huge birthing in the soil. And that's really when kind of enough plant matter had accumulated that they had a whole new task open to them, which kind of gets me excited because we have like quite a bit of pollution, and so I'm hoping that there will be a new order of mushrooms that basically just help detoxify a lot of stuff like water or soil, rather than breaking down kind of wood and peat bogs into renewed resources. They'll be able to do that maybe in the air or water with petroleum chemicals, which is not something that's entirely unheard of, especially with their ability to kind of reach through their DNA and turn stuff on and off. Um, so these are eugarics, and these are also a part of Agaricalis, which means they're bearing spores. And so they have club-like, they have all kinds of different forms, almost like a coral brain. I think this one might be resupinated. Um, club-like, I forget these. But I know this is a bird's nest fungi, and these are very, very small, and those are each of its spores, and basically it waits for a water droplet to land and splash them out. I found these quite often around the campus. They're absolutely beautiful. I've seen some with almost like a pearl blue color of the spores on like a golden copper nest. Very, very pretty. Um, but yeah, you can also get like white rot inside houses. Some mushrooms have even kind of fruited in the corner of bathrooms. There's like a whole subreddit about those. Um, so boletes. So these are also known for having like a pore surface, and rather than the gills, they basically bear their spores out of a pored surface, kind of like what you see on a polypore. Um, and so these do include the puffball, the earth ball, I guess, and toothed and gilled mushrooms. And so you have white rot and then you have brown rot. And I forget exactly which is which, but one is breaking down the lignin and one is breaking down the cellulose in wood. So they do feed on two different things. And most are mycorrhizal, but some of these are mycoparasites.
If you want to know which is which, like when you're walking around in the woods, the white rot fungi turns the wood all spongy, and the brown rots turn it all cuboid. And I don't remember either which is doing which in the wood. But that's the way to tell if anyone's curious about that.
And then the Rusolailies, if I'm saying that correct.
Yeah.
And these have some beautiful pictures. And so he says they're probably the most morphologically diverse. And so a lot of these have teeth, basically, or it can be like a club structure, a polypore structure, almost a jelly type, gills. And these ones basically are saprobes, they're ectomycorrhizal, root parasites, which are pretty common, and then a lot of insect symbionts, which basically a lot of the, basically you'll have a beetle that has a little pouch on it. And basically the spores or even active mycelium will basically live in this pouch on the back of the beetle. And so as it crawls and as it eats through the wood, it's basically leaving behind a little trail of mycelium that will then spread out and begin to decay it. And typically a lot of those beetles will then eat the growing mycelium in the hole that it's bored, and basically just create a little home where it can eat food, be provided shelter, and then the mushroom can also eat and have food while basically feeding the beetle. And so polypores are in the Agaricalis, or Agaromycetes, I mean. And so these are many, but not all of the bracket fungi. And so here, I believe this one's a Reishi. I'm not sure if that's another Reishi or if it's something different. Sometimes they do get that very, very black top, but I've seen it with a couple other things as well. Obviously, I believe this might be like a honey mushroom, or maybe even, or not a honey mushroom. I'm not sure what that one is actually. If it's
Those look like oysters to me, or maybe
that's what I
they're
was going to say.
the
He does say, but that's
Gopheles, the maitake, and I think the one above, I think that's the foamies.
Oh, okay.
Yeah, I'm just like looking at the slide here, and I think, yeah.
Oh yeah, Fomitopsis and Ganoderma. So yeah, that one would be this one. And then I'm guessing this one might even be the
Looks like the
dog bark.
cauliflower, yeah.
I know we've seen a lot of dog bark fungi in our compost lately. I feel like I've been seeing it quite a bit, especially out at Purgatory. But the Aurucularis, probably not saying that one right, but that's the woodier. And like I was saying, they're highly, highly cultivated in Asia. It's often called the black fungus, but these are used in a lot of soups. They're very, very good in like miso, and provide basically a whole additional nutritional component. They'll soak up broth very, very good. They're mostly they're like 99% water, or well, that's probably an exaggeration. But they're vastly composed of water. So when they're dehydrated like this, and you add them back into a soup or a broth, they pretty much just absorb that flavor, which is like a pretty cool culinary thing. And then the chanterelles. So these were kind of early diverged. But these ones often have like gills inside them, but they can be toothed, clubs, or even crusts. They're also mainly ectomycorrhizal or saprotrophic, meaning they are basically like feeding off of something else. And obviously just saying it's very economically important for foragers. And I don't know if that's been cultivated before or not. I don't think most ectomycorrhizal ones have been, although I'm pretty sure that India does have a culture of it on agar that they will send out. I'm pretty sure that one's covered in the showcasing ectomycorrhizal fungi paper, if anyone was here when we talked about that one in the old talks. But so here are the Gopheles or the phallales. And basically a lot of these are your stinkhorns. They produce basically a sticky gleba, and that's what contains most of their spores. And so basically they attract flies as their pollinator. And then the flies basically get stuck to the sticky gleba that smells profusely. And then they basically fly off and carry their spores. And if you know anything about flies, you know how often they're basically flying around and landing on different things and checking different things out. So it's a very, very effective way of spreading your spores. I've also noticed butterflies really kind of like a lot of the, not only the Gopheles and the stinkhorns, but also just a lot of different mushrooms. I've really seem to notice a lot of butterflies landing on them if they have the opportunity to. And so here are the Gastraelis, and these are the ones like the Texas Star. And basically, these ones produce basically gaster means like stomach. And they create this pouch with their spores in them that any type of pressure, even air change pressures, will basically shoot their spores out, kind of like a cannon. And then here's kind of when they diverged. Although I think he's pointing out that they're kind of spread throughout a bunch of different trees, or they're not taxonomically condensed, I think, as he would probably say it. But basically, there's a lot of unknown evolution in between a lot of the different species.
Yeah, that's the convergent evolution that I was talking about. I remember hearing a really nice talk about it, and I really wish I could remember who was giving the talk, but at the Telluride Mushroom Festival about
Nice.
this like convergent evolution. So it was like someone presenting on like a paper that they were publishing or whatever. But
Nice.
yeah, it was really, really, really interesting to think about. It's like how much these techniques just worked, and like yeah, it kind of obscures some of these relationships that like you'd think
Mhm.
just by looking at it, oh, these things are related, but you know they're definitely not. And similar things going on with like things that look
Yeah,
like
yeah.
something that grows in Europe and it got named in Europe, you know, a
And
long
then we
time
call it the
ago.
same thing here. Yeah.
Yeah, and then they grow in the United States, and they're completely different species, but even though they look visually identical, but they've just
Mhm.
been separated through geography for so long that they're different species. So
Well, and I know in particular with the Rishis, because like Ryan Paul Gates talks about how like a lot of I believe you're saying a lot of the Asian papers were really meticulous about categorizing like the spores and stuff of any of the Rishis that they worked with. And then someone went back and kind of realized that the descriptions of the spores are not consistent among basically the American strains and the Asian strains. So basically saying like, even though these are all called the same Rishis, I believe it was for Lingzini, he was like, these are actually two or even maybe three or four different species of them, having two species separate out in the US and Asia, and then even two more from those ones. Because I believe he was saying the ones that we have in Utah are like a very unique sect of them that don't match any of the spore samples of the other American Rishi Lingzini. So really crazy stuff.
yeah,
And so then explosive, I believe these ones are the bird's nest, and that's kind of when the water lands. But I do think there's one that creates some sort of burst. Um, I'm not sure what will happen if I click on that link. uh, Daiquiri mycetes. So these are the brown rots, and they're basically like a jelly mold. And they have the forked basidia, which means so if you put them underneath a microscope, they would have like the tuning forked basidia. They would have basically two sets of Ys coming off of it. And they're yellow, gelatinous, or cartilaginous. So like almost like cartilage, very kind of like tough jelly. The Tremolomycides. So that looks like it's another type of jelly. I don't think I've ever seen a clear or a white one. But basically, it'll also grow molds on other mushrooms and human infections as well. But it looks like these might be pictures of them as yeasts. Yeah. Oh yeah, and some of them are just yeasts. And so now we're moving into the Utsinolog Mytina. So these ones are like the corn smuts, and they produce teliospores. So these are very, very thick spores that these ones have. And they're mostly on grasses, so like corn or wheat or things like that. Here's the whole life cycle of it, which this one's pretty standard. Some of them, I think he goes into actually have 2 different hosts that they have to utilize. And so this is actually pretty cool. These are called Apresorium. And so basically a spore will land on, say, like a leaf structure, and then it will basically germinate a little peg, and then it's basically searching for a weak spot in the leaf structure. And when it finds one, it'll basically begin just growing and forcing down into through the cell wall of the plant. And so basically it has like a little pin almost, and then it just puts all of its growing force behind that. And then once it reaches into or beneath the protective skin of a plant cell, it just basically begins eating and germinating into all of the other cells of the plant. And so Historium is basically like these little circles. And those are different from arbuscules, which are in ectomycorrhizal fungi and things like that, that are basically allowed through a root cell wall or something like that. They create a Christmas tree-like structure inside the cell. And that structure both gives and receives nutrients and chemicals, whereas the Historia only takes. It only is able to absorb, at least that we know of. It could be releasing chemicals to help kill the cell or something, but I believe most of the time they only absorb, and it's kind of this parasitic life. And so these are kind of diseases that can get on both animals and people. They're highly susceptible to zinc. So if you've ever used Head and Shoulders, that's basically a zinc fungicide. And then the Puccino Micatina. So yeah, these are the ones that have 2 different hosts. So part of their reproductive cycle is done on wheat, and the other part of it is done on a barberry. And so, but one part of it will repeatedly infect wheat, so that it can basically create a lot of, I believe, like asexual spores, and then it goes to produce sexual reproduction on the barberry tree. And so I believe, okay, so these ones are rust. Here's the Puccino gramus. So that's rust wheat, rust of wheat. Here's rust on brambles. And then I believe this one is on ash junipers, and then this one is the apples. I've been trying to find this one on the junipers in our area, and I've never seen them. And I think that's because it also requires the apple tree for the other part of its life cycle, because I believe this is the one species, and it requires both life cycles to jump back and forth. But man, I would really love to find like a nice orange tendrily blob like that out on one of our juniper trees here.
I've seen one in College Station area on my.
Oh,
my
really?
parents',
Nice.
yeah. So maybe, yeah, I don't know what it is. Maybe you're right about there not being the right host for around here, and I don't know if the one that I saw was specifically the one that was like pictured here, but
well, and
they're pretty cool looking.
Whenever you have 2 different hosts, it really kind of makes me wonder if there's not a second, like a substitute host or anything like that, because obviously that thing has like a very, very diverse genome in parasitism. Like if it can attack 2 different trees, then who knows how many different trees it really can attack.
Someone just said in the chat that
Oh, it's
it's on
edible.
Wikipedia that it's edible. I mean, I,
I would try and taste it, maybe.
I know jelly fungi are mostly edible, and yeah, they're just kind of like a texture thing that if you can get over that, I think you'd probably be fine. But sometimes the slimy gives people trouble, especially if you're not used to it.
Yeah.
Yeah, I wonder who was the first person to try eating it. That's someone who was hungry.
So, and I think these are, so this is an auger plate, or I believe it is maybe chrome auger, but maybe not. Oh yeah, salmonella color, or salmon color. So basically, this is a yeast that when it grows, it pigments, I believe. And I know you can use chrome auger to determine different types of candida. So like basically, you have candida all over you and in you. It's mutualistic or symbiotic fungi, depending on where it is in your body, I believe. But like we have it in our mouth, we have it in our stomach, we have it on our skin. You even have different species on your skin of like your armpits and belly buttons versus your like hands and feet. It's a pretty crazy one. And most of the different colors will appear on chrome, or most of the different species will have a different color on chrome auger. Here are basically some on moss. And I think, I'm not sure if this is a spore or what that is. But those look to be like growing tips off of a moss. So this is kind of just talking more about the complex life cycles. And so here's like a microscopic picture of them beginning to bear spores out. Here are the telospores. So here it is on a new leaf, and then it creates a very similar structure. So usually whenever you see an area exploding out of the skin like this, and then a large different colored mass underneath it, a lot of times that's very common for what I've seen in a lot of parasitic fungi. Especially if you have just a bunch of basically almost like a little volcano of dots, or basically like a big urn of dots. These make me think of truffles a whole lot, except a microscopic truffle within a plant cell. And so it's really crazy that they can basically affect completely different parts of the plant too. Like several of them will grow on leaves, and then the actual like corn or other parts of the actual like seed part of the grasses will be affected. And so that is basically the three major ones in Basidia mycota: mushroom, rust, and smuts. There's also Ectorrhizomycetes, which are strictly root parasites, and Willemia I do not even know what xerophilic means. I'm guessing that's like extreme.
I think it's dry loving
Yeah, you're right.
molds.
Because like xerolons, yeah. So molds that survive without water. And then that is the end of all of the Basidia mycotas. So that's all of the Basidios. There is also Ascomycota, and I believe he even has spores. Which, if y'all want, I believe the spawn is ready now. And if you want, I can show you how I bag it up, or I can show you how I rehydrate substrate. Or we could do, I mean, I think it's already been an hour. If y'all want, I don't really care. We can go through a whole nother lecture, or whatever everyone's feeling. It's community day.
Does anybody have any questions about the presentation that they would like answered? I love talking about evolution. I think it's so fascinating.
Me too. And there's just so many different mechanisms going on, especially in the DNA, like the different promoters and like basically things that are just proteins that are in your body that either turn on or turn off parts of your genome. Um, and so like I get really excited too with proteomics because I think that's like learning how to read and write the code of DNA. And I mean, I think that it's very possible soon we'll be able to just run the DNA of a mushroom and determine if it's edible or poisonous, or even its medicinal values. Um, but we just have to get a little bit farther along with our genetics, which is happening day by day very rapidly. Um, William Padilla Brown's been doing a lot of like not necessarily low tech, but much more accessible versions of it as well.
Yeah. Well, that's just kind of like a general trend.
Mhm.
One that like kind of noticed in like, because I make music as a hobby and stuff like that too. And it's very similar where like the development of the technology just makes it more and more accessible. So like now, for music, it's like I have, you know, a really nice, more or less a really nice studio in my house, but it's really all like software stuff. But like
Mhm.
back in the day, you would have needed to have like a whole building with like a bunch of electronic equipment, but now it's all like the software
Mhm.
emulation on it's really good. And a similar kind of development of like the like the PCR technology and some of the gene analysis stuff is the further it develops, the cheaper it gets, the more accessible it gets to people who don't have a lot of money. Whereas like it used to be
Mhm.
the only
You have to have a
institutions
lot.
with enough
Mhm.
is universities and like big libraries and governments. Basically those are the only really big ones, uh institutions large enough to like be able to fund that kind of research. And now it's in a way it's like being democratized a lot. Obviously like there's still barriers,
Mhm.
through certain levels of finances, but it's changed a lot. And I think it's more accessible for just like quote unquote lay people to kind of get into this stuff, if you're dedicated and don't mind reading a bunch of books and learning a bunch of new jargon.
Mhm.
Basically all colleges anyway.
Yeah, which it's also pretty neat that like you're saying, like it would take really large institutions and really large sizes as well. And both of those things have come down, both like the cost, the amount of room that it takes up, the amount of like power you need to do it. And like it seems like with every new advancement, it gets even faster too. And I think that people are really kind of starting to see a demand, especially like, well, and almost kind of like you were saying, creating a demand. Like if there was a way to be able to sequence the genome of any mushroom while you're at the grocery store or something like that, I mean, if you told people they could do it, I know that some people would. Um, and so that's really cool to me too, because I'm like, if um, if we can collect that much data on mushrooms on an area where we don't have any data at all, that really kind of like opens up a whole new realm to us, both in like food and medicine, and in kind of just reconnecting with our own wild foraging, and things like that.
Yeah, totally.
I get really excited for it.
Another thing that I really love about evolution is just kind of the thought that like nothing and it's like kind of it's like correcting kind of a misconception that people have through like talking about evolution. This is another thing I learned at the Telluride Mushroom Festival, so shout out to Telluride.
Mhm.
um but like it thinking about evolution as like a web of evolution, or like as like a tree with branches and tips, as opposed to like what most people think of with evolution, where it's like steps along a
Yeah.
trajectory. And so when you look at these organisms, like they are just as evolved as humans. We tend to like we as like mycophiles really appreciate fungi for their amazing abilities and stuff like that. But like, and we call them advanced and all these things, but they've just been evolving as long as mammals have and humans have. And it's
Mhm.
interesting to think about that, like um, when you look at fossils, they are just organisms that have stopped evolving. And so like that's kind of how we
Mhm.
date them. It's like when their evolutionary, these species stopped evolving, or when these lineages stopped evolving. And like what we are living among and with with today are is highly advanced as we are, even if like the branch of their tree like diverged from ours a long time ago. But like and now I'm going to get a little political. But like when people like the conservative line about life starts at conception, and it's like, no, life started like 3.5 billion years ago, and it's been an unbroken chain ever since. And like maybe we should respect that and like deal with climate change and stuff like that. It's like such a like that. To what blows my mind about that is like that is the reality.
Mm-hmm.
And so like everything else we kind of is like a kind of a fancy little story we tell ourselves about things, but like the reality is we are intimately connected and have been growing up with all the organisms around us since before, like, you know,
Yeah.
like life first started, DNA and RNA first started replicating in primordial ooze due to like volcanic lightning or some shit like that, you know. Like it's been
Well, and
an
the
unbroken chain since then, you know, been disrupted but still an unbroken chain.
Well, and the coolest thing to me is like is the recognition of like life with other life, and that like nature is almost always trying to form a community. Not always, because you do have pathogens, you do have like really intense like lone predators, but for the most part, like almost anything in life has a symbiote or has something working with it. And I mean, like we tend to think of like the macroscopic a lot of like, well, I guess like we do have a pet or something like that. But like you have millions and billions of different species of bacteria, of viruses, of fungi, all inside you, you know, in your mouth, in your stomach, in your skin layers, that are all providing different benefits for you. And it's like each one of those evolved to take care of that part of you. And so like it's really awesome to me that I just see that like especially in the kingdom of fungi so much cooperation. And it's like there's also a lot of endophytes that literally just they're in every single cell that you have and they're just waiting for that cell to die so that it can start decaying it. And so just like some very very beautiful relationships that nature has given birth to through evolution.
Yeah, totally. The endosymbiosis of mitochondria
Yeah.
into eukaryote cells and also into plant cells, the chloroplasts and stuff like that too. It's like, yeah, fractals, man, fractals.
And I was gonna say, I think the spawn is done. Let me see if it is, and then I'll try and show it.
Yeah, for sure. And I will take this opportunity to do a little plug. Yeah, so if you enjoyed this presentation tonight, consider becoming a member of Central Texas Mycological Society. You can find a link in the description below that takes you to our website. Yeah, it helps support presentations like this. And as now that COVID seems to be kind of lightening up a little bit and people get vaccinated, we're able to start meeting in person again. So we'll be, we're probably in the fall, we'll be doing a nice big foray where we go out and hunt for mushrooms in the woods, and maybe do a little camping or stay in some cabins or something. Haven't hammered out those details yet, but yeah, so that'll be probably a members only thing just because it's been a while since we've been able to do anything like that. So if you're interested in becoming a member, yeah, sign up on our website. We also do, um, we recycle old mushroom spawn blocks from some of the local mushroom farms around town, and you know we keep that mushroom spawn from going into the landfill, and we help distribute it out to members and community and in just into the community in general, for people to use in their own home cultivation and get their feet wet and growing some mushrooms, compost in the garden that makes lovely additions to your compost. Usually you can get some mushrooms out of them. You can use it to seed um, a mushroom bed. You know, at that point, the world's your oyster mushroom. So yeah, head on over to the Central Texas Mycological Society, Central Texas Mycology.org, uh, for more information on that. Yeah, like and subscribe the YouTube, comment, engage it, share it with your friends, ring the bell if you want to be notified, all the YouTube stuff that everyone is indoctrinated into now. Yeah, thanks for listening. You ready, Ryan? Looks like almost. Yeah, evolution is something that when I get to stargazing, it really blows my mind. And working with mushrooms is a pretty good way to kind of get a new appreciation for other organisms and whatnot, just because they're so different from plants, different from animals, obviously, in a variety of ways. And I think plants tend to fade in the background, but mushrooms can kind of help break people out of that stagnation, I think, especially if you're getting into foraging and stuff like that.
So here is pretty much what it looks like after being hydrated. So I mean, that was all within our talk. It was literally just one Instant Pot bowl of birdseed and water. And then I pretty much just scooped these into spawn bags.
Hey Ryan, can you take off the screen share so they'll be a little bit bigger?
Oh yes, absolutely.
Awesome.
Sorry, I am not used to using Zoom. um,
So earlier at the beginning, you showed us how to hydrate it and get it ready for sterilization, and you're about to bag it up, right?
Yep.
The process of mushroom cultivation. Live for everyone to see. Let's do. Anybody got any questions about evolution? No.
And I usually just use like a coffee cup, just because it's really hot.
Yeah, so the first step was steaming it in the Instapot just to force a bunch of water into it and get it ready. But I don't think that was the sterilization. I think then he's getting it ready to put into a pressure cooker. Right? Am I right, Ryan? Or
Yes, that
you
is
got a different
correct.
method? Cool.
So yeah, basically once you get into a bag, you fold it up. I usually tape it too, just to be. I just don't like any water getting in or out while it's in the like All American. But basically, you just roll it up. Most people put rubber bands or they tape it, and then you just load it into a pressure cooker. I'll take y'all out to the current one. It's actually got several bags of substrate in it right now.
Why do you prefer brown rice or not brown rice, birdseed over like rye grains or something like that?
So I just haven't used rye grain a whole lot. I've used oats quite a bit, and they tend to get some fungal activity. Birdseed does as well, but I feel like it gets a lot more bacterial. Same thing with popcorn. I feel like they have just more bacterial ones, and I feel like most mycelium can deal with bacteria better than fungal. Plus, it's like less of a risk for moving it around. But this is basically the sterilizer, and it's an electric one. So you basically just turn it on, set a temperature limit, and you burp it every once in a while. Here are propane ones, which I've been trying to not use as much, mainly just to switch over to electricity. It's just terrifying having propane running in your garage.
Yeah.
And then these are basically some of the bags of substrate. And so, like I was saying, I tape them up. The black lines there indicate this is autoclave tape, so it shows that it's reached sterilization temperature for a long enough time period. And then basically I'll open these up and dump spawn into them after it's been colonized. And then these will make fruiting blocks. Kind of a video of what some spawn blocks look like. So there are some Rishis fruiting. But we have like king trumpets. Here's some lion's mane fruiting. These are some pink oyster fruiting blocks, yellow oysters, chestnuts, a few more Rishis. The Namicos had a much harder time with the environment. So I only have 2 bags of those. But these are all things that I'm hoping to kind of do some fruiting trials of, especially moving them over into substrate. Because these are ones I haven't seen a whole lot in gourmet cultivation. So really kind of trying to find ways to get them out. And then if anyone is interested, we do have quite a few mushroom cultures. And like as a member, if you ever want a copy of a culture or anything, just let us know. And we'll basically make a duplicate for you. And we're trying to not mail as much as possible. But if the only way you can get it is through mail, we can definitely mail it to you. But so yeah. And essentially what I'll do is once that bag of grain is cool enough, I'll sterilize it. And then basically put some of those bags. I usually get about, if I do 2 Instapots, I'll get about 10 bags worth of 3 to 4 pound bags. And then basically I'll just take all those ones in the tent. I usually punch them and break them up pretty good. And then mix them with the fresh spawn that's just been sterilized. And then that's how you start a new spawn bag going, so that you always have that in inventory. And then the ones you want to move over into a fruiting bag. The fruiting substrate really just kind of helps keep, oh, the air conditioning in the garage? No, there's no air conditioning. Sorry, I just saw that. And it is steaming in the Instapot. But basically, you can move these into substrate bags, and that basically just helps provide like a woodish barrier, basically a higher carbon source than most organisms can eat, to protect the like starch and the other nutrients in the spawn. So one, it kind of decreases the amount of mushroom flush you'll get out of it by separating out the spawn. But that's good because you'd probably try to get like 8 or 9 or 10 flushes out of one bag, and eventually it would get contaminated. So basically it's just a way of spreading out the food and putting a little bit of protection over them, so that you have a lower chance of spoiling a whole meal as opposed to like a side dish, if that makes sense.
So you're growing all those different species on the birdseed, and then you're transferring it to like a wood sawdust substrate or something like that. Like, what is your the term is bulk substrate that you're using?
So my substrate, what I usually do is half of hardwood. And I mean, I get a bunch of different ones of that. You can get the mushroom media online. Hardwood, I typically just go to Walmart or like HEB or Tractor Supply and get like competition barbecuing pellets, the fuel wood pellets. And those are pretty much just compressed together. So whenever you pour, I do boiling water. If you pour boiling water over it, it expands almost immediately. And so I usually do about 2 pounds of pellets and then like 2 pounds of water. And that'll usually flush out into several different bags.
Cool. Are you supplementing nutrition at all?
So I haven't been. But my recent auger experiments showed a lot of mushrooms liking the compost or the organic fertilizers, and they're just hydroponic fertilizers that I added to the auger mix. So lately, what I've been doing whenever I do the substrate mix, I'll actually add the fertilizers to the boiling water before I pour it over the substrate. So that way, all the substrate is absorbing basically like fertilizer water into it. And that makes them kind of take to the substrate a lot faster, and really kind of like makes them take over it very quickly.
So you're just using like an NPK
Yeah.
organic fertilizer? Wow.
And it's like one of them is it's called Black Diamond. It's from General Organics, but their Black Diamond is like fluvic acid. So it's specifically formulated to like grow or to like encourage mushroom growth. I believe it's humic and fluvic acid mixed together. And I think they unfortunately mine it from like the peat bogs in Canada, or from the boreal rainforest in Canada. But it's definitely like a very, very rich carbon source that the mushrooms love. And they grow a very, very thick growth over them as well. And then I also add to that just kind of their general nitrogen one that's like a 333 of NPK, I think. And I normally just do that because I've seen them react very positively to both nitrogen and phosphate fertilizers. I think that's something that a lot of organisms, if they can take it in and use it properly, it really kind of fuels their internal engine.
Yeah. I guess I've just never heard of like using plant fertilizers in this process. It makes sense that you would, but like, because like I would always just use supplement with like wheat bran or something like that,
Mhm.
sawdust and wheat bran, and that worked pretty well when I was able to cultivate more. And I'm looking
Um,
forward to moving because I'll probably be able to get back into it, have a lot more space.
nice. But yeah, I was gonna say I've even been trying to when I pressure cook the spawn, sometimes I'll mix in, and I'm literally only putting like 5 to 10 milliliters of the fertilizer in, and especially the black diamond, that humic acid one. I mean, I think that's really where the power is at. And I mean, even just like a few drops of it will turn water almost black. Like it's a very, very potent carbon source. Um,
It's got me thinking of what you could do just like with compost teas and stuff.
Yeah, exactly. And like you're saying, it's something that like no one has really kind of mixed together. And really, I probably wouldn't have if I wasn't focused on hydroponics and then taking a mycology class. And then once you start to see a lot of the soil-borne fungus, especially like the ectomycorrhizal ones, they're obviously using those same fertilizer chemicals in their growth, or at the very least, they're using them to trade to plants for other sugars or something like that. Um, so I've really kind of gotten interested in doing like a cocoa core soak with them for like a monotub experiment, and see, because I would really like to start culturing ectomycorrhizal fungi in tubs, because there's obviously just something the plants are adding that if we can really get that down and add it ourselves, that will really unleash a whole new cultivation era.
I mean, I feel like you could also go just like figuring out ways to guarantee inoculated plants. Well, fruit
Yeah.
mushrooms would be another great way to go to with that too.
Um, which, yeah, I was going to say, I think in that showcasing ectomycorrhizal, or at least I know with the Thailand boletes. That's basically what they're doing is potted plants in greenhouses. And they'll basically just tear up mushroom spawn because they can get the spores on auger, they can get the auger onto spawn. Then they'll take the spawn and mix it into like a bedding substrate and plant the plant in it so that when it grows, it's been infected with the ectomycorrhizal ones. Um, and so that's something that's super interesting to me too, is because then, especially like trees or really anything, if you get a large enough pot, you could continually harvest off of. Um, and then you're also really kind of in control of the water and the nutrients, which means you kind of have this ability to ramp up the cycle really fast, if you wanted to try and increase production.
Yeah, and if you, because I think a lot of that stuff is also uh, climate control too. Like, or not
Hmm.
climate control, but like you would have the ability to kind of, if you're doing it in like a greenhouse or whatever, with the potted plants, you'd be able
Okay.
to just trigger those climate conditions that they need I'm thinking about chanterelles right now just because like some of our members have been finding them this season, and it's been really cool. But we've just been recently getting a lot of water.
Um,
You want to
so
go ahead with that one?
I don't know off the top of my head. Um, I'm trying to think. I know a couple of them are trees. Let me see.
I bet most of them are trees of some kind, and then potentially you could have like a polyculture of the fungus and a tree. The one that pops into my head that's like that already exists, in a more or less commercial scale is like pecan orchards with pecan truffles.
Yes.
Except most
Yeah.
people aren't going out and harvesting their pecan orchards for truffles and trying to sell them and distribute them because people just don't really know about them around here. So,
Mm-hmm.
but I think you could, yeah, I think you could do a lot of stuff like that. Or like if there's ways you could figure out to inoculate even existing trees, you could maybe even protect them that way by like
Mm-hmm.
without short of abolishing capitalism, basically you have to find ways to trick people into thinking things are economically valuable. So if you could trick someone into thinking that because the mushroom, if you could harvest mushrooms from the tree and that was more valuable than cutting down the tree itself,
Mm-hmm.
you save the tree that way. Um, I'm for abolishing capitalism. Barricades. Anyway, I digress.
I was gonna say I do know that the boletes, which it was the black tropical bolete, they were cultivating them on sorghum spawn, and then they were moving them to rubber tree substrate basically as their like wood finisher. And then they were able to actually get fruits out of their bag of the boletes without having a living host that way. So basically, possibly even taking up the hosts and chopping them up and using them as a substrate, which would again kind of be an economic value. But I mean, I hope we're not doing it in a whole bunch of bags at least. I would hope that would be some sort of like outdoor long bed pile or something.
Yeah.
But I mean, definitely a way to, because like I was saying, the plants are releasing or producing something that is the limiting factor. So if you can find a way to get that limiting factor and get it into the substrate in the spawn, then I mean, theoretically, any mushroom is cultivatable. Thank you. And it is, luckily, most of my mushrooms did phenomenal through the winter. And I have not had really a long Texas summer with it. I have noticed definitely a lot of fruiting kind of dying off quicker, a lot of kind of senescence in some of the pinks up at the top, and a few other things. But yeah, I'm really kind of holding out. I don't want to get a swamp cooler for the garage, but I might have to.
Well, the problem with swamp coolers around here is we have such a high humidity
Maybe already,
that they
yeah.
don't really work that well. So like it gets to be really hard to grow mushrooms around here in the summer because you're constantly having to like cool the building, and that usually means running an air conditioner that dehumidifies the air,
And
but
then
then you got to
reheating.
humidify the air, so you're constantly fighting yourself. So
Yeah.
I think it can be done, and I think there's some way like I think doing things in a seasonal manner and like changing your crops around, I think you can do a lot with like shade houses and like misters that way, because if you can keep it around, like even on some of the hotter days like in the 90s, like you probably still be able to get some mushrooms to fruit if they're like in a deep 80% shade in like a greenhouse, and like but that's kind of me more speculating and like just spending time in some of those places being like, well, if you had the right mushroom species and like maybe even potentially something that was like um, a native around here, that would also help too.
Well, and I like you're saying native. I've even noticed a lot of similar species that are just different varieties, um, have vastly different temperature and humidity ranges. That's why I get very excited about like local Texas oysters, because that one's going to be able to survive the heat, because it has been. Whereas like even if you get a pink oyster or something that's known for some heat tolerance, but it was like wild forage from up north. You don't really know, it hasn't really experienced kind of those high heat temperatures. So you don't really know how well it'll survive that.
Right. And one of the things that like is like finding so we're like in this weird intersection of bioregions. Like we get a lot of tropicals that grow here and stuff like that too. And so like I think I know Angel, one of our members, she's been really, she shared this the other day, and she actually cooked some, but of the Favolus brasiliensis, which is like eaten by the Yanomami people down in Brazil, but it grows in like abundance up here, and so like that's also another avenue potentially even for cultivation. And like I've seen that fruit in relatively warm conditions, and it's just a matter of like getting people comfortable with, like you were talking about earlier, just like new wild foods and like
Mhm.
incorporating those into our diets and stuff like that. And so yeah, I think there's definitely a lot of room to grow there also.
Which it's awesome because there's so many mushrooms out there, and I mean, in my opinion, like I was saying, they could all be cultured. I mean, it would take effort, significant effort, to figure out how, but that's not what outside of our reach by any means. But then the flip side of that too is making a market for it, or convincing people that they are not dangerous, because there is a lot of stigma kind of on mushrooms. Even some people that I've given mushroom blocks to, or helped them kind of inoculate their own bags. They're like, they'll ask me like, is anything going to fruit out of this that's going to kill me? And I'm like, no, like a wild mushroom spore is not going to take over your bag and like produce, like the destroying angel or anything like that. Like the only thing that's going to get in there and eat your mushroom that you want to grow and eat is going to be like a mold. And like most of the people are in ag or something like that. And I'm like, what mold looks like if you've even left food out on your counter. You can identify what like green molds and black molds look like. So if you see a patch of that growing in your bag, I would probably remove it. But like that's not going to fruit into a mushroom that you mistake for lion's mane or something and then eat. Um,
Yeah, totally. This reminds me of a funny story though. This is a lab accident that happened to one of my friends on their farm. Some got some really bad contamination from another culture that they had in their lab, and they
mhm.
didn't realize it till they like had mailed out a couple blocks, and then they're like, "Did you mean to send me a turkey tail? Because I got a turkey tail." It's like, oh, what? Something happened here. They accidentally they were growing only all turkey tails on accident.
Oh
So
wow.
yeah, it was kind of like a weird fluke or something like that. And I think they had to kind of like clean everything, but and kind of start over on their production run. But
That
yeah,
is a pretty crazy story then.
yeah,
But yeah, which I mean, life finds a way, and they say that like I know like one of our facts in mycology was like every breath you breathe in is 10 fungal spores, or I think maybe that was in Fantastic Fungi. Um, but so it's like they are proliferous, they're everywhere. And the spore dispersal is like insane. They can go very high up in the atmosphere and make it very many miles before kind of coming back down and settling. And so like it does make sense that like half of the job of the flow hood is to get rid of competition.
yeah.
Um, but or at least like the fungal competition.
Yeah. So speaking of flow hoods, how do you do your inoculations, and like where do you use a flow hood, or are you working in like a cell air box, or what? Yeah. What do you prefer? What do you recommend?
So here I'll actually take you back to the garage real quick. Um, but I basically have a fan filter unit, which is basically just a little bit cheaper than a flow hood. I was kind of looking at the prices of them, and flow hoods have really exploded over the last year and a half. Basically, when the lockdown happened, a lot of home mycology exploded. And so, like within that time, Paul Stamets's fungi perfecti flow hoods like increased by like 100%, like pretty much doubled in price. Most of the ones that I could find online from like Instagram or Facebook or people who homemade them were like equally expensive. You're looking at like $1,500 for like a 1 foot by 1 foot flow hood. So basically, what I got was this is a fan filter unit. And basically what it does is it's supposed to go on the roof of a medical facility. So I kind of had to like wire it up myself a little bit. But basically it has, I would say, probably like a 1-year warranty, and is like $700. Whereas the flow hood, at least from like Fungi Perfecti, to get one of this size, I think is like $4,000. And then the filter that you swap it out with, which they recommend to do yearly, is about $500 or $600. So I was like, I would rather just rebuy a fan filter unit every year if I had to, versus pay a high initial cost for something that I'm still going to have to replace an expensive filter on anyway. But what I started out with is basically what I call a faux hood. And I think Willie Myco has a few videos on how to make it. And basically it's just like, I have 2 of them, but it's basically just a home air filter. And this one, I've got one on the front and one on the back of the actual fan itself. But I'm just one on the back is sufficient for most people, and really kind of helps increase your airflow. But basically it's just like Gorilla taped air filters to a box fan. So it's a very, very cheap way. And as you can see, I kind of keep most of my stuff in a corner here between like my faux hood and the actual fan filter unit, just to create like a corner of very, very clean airflow. And so that's what I did. I started off with a still air box, but the home filters on the box fan significantly, it made me feel a lot more comfortable, and it worked a lot better than I kind of anticipated it to.
Yeah, that's cool. I've never seen that before, but that's seems pretty ingenious.
But yeah, so that's normally where I do most of my, and I'm hoping to set up a second tent that's a 10 by 10 in there. And that one will basically be a clean room for working. And then the other one will contain spawn and like kind of fruiting box.
Cool. What kind of fans are you working with in your fruiting room, or are you doing any of that? And how are you humidifying that space?
So actually, I'll just back and show you. I've got an inline fan. I probably should have just turned around and showed you. I've got an inline fan that is kind of built for like guerrilla grows for cannabis to export the air and carbon filter it. But it's basically just a inline fan that hooks up to this sleeve right here, and will basically exhaust the air out. That's pretty much the only fan I have in it right now. And then I use a very simple, it's a reptile humidifier. And basically I just run it up through the top and over to the bags. And then if you can see in the back, it's hooked up to different ink birds that turn it on whenever the humidity drops down, and turn it off whenever the humidity gets back up.
Cool.
And then I really don't have that impressive lights. They're just old fish tank lights going. And I use those just because they have an automatic 24-hour cycle on them. So they'll get brighter throughout the day and they'll change shades. And then they'll basically go to a blue at night. And they'll even have a moon cycle in them as well.
Neat.
But yeah. And then if anyone is interested, so this is like my spawn and substrate corner. So I do use a lot of corn cob bedding. It's been very, very hard to find. Tractor Supply stopped carrying it. But I use that in my substrate, as well as kind of like competition wood pellets. And these ones are charcoal, and I've been trying those out just because I've been kind of wanting to see if that affects anything. I also have a fruit tree blend to see if kind of like a sweeter wood has any effect on it. And then I normally just use horse oats, chicken scratch, or wild bird seed. And sometimes I'll mix those together just to get a good blend. Sometimes I'll just use one. Really, I'm just kind of playing around with them to do different experiments.
Cool. Do you mix up your substrates kind of frequently to keep your spawn interested? or do you?
So I haven't run too many from different substrates, or like fruited off of a substrate and then going to another substrate or anything like that. But I do change up the substrate quite a bit. I've been doing the soy pellets from the mushroom media online, and then mixing soy pellets and hardwood. And then I also recently got a wheat bran bedding. That's basically like a vermicompost bedding made from wheat bran. And so I'm trying to basically supplement that one in. Um, but yeah, most of the time it's just hardwood and corn is what I really like to run with. Just because I feel like most mushrooms eat corn very effectively, whether it's like corn syrup in Caro, or like popcorn as spawn, or corn husk as bedding. And I try to keep a little bit of all of like corn somewhere throughout the line. So that way, at least no matter what they're going to, it has some familiarity with it.
That's a good idea. Yeah. They must love corn like we love corn.
We grow a lot of it, so
Yeah.
that could also be part of it.
It makes sense that it would end up in that kind of stuff too, just because, like you said, we grow a lot of it.
Yeah, well, and it is very like, I mean, it's a sugar packed, it's an energy packed grain
Yeah.
for sure. But yeah.
Well, cool. Do we have any questions in any of our chats here? The Zoom chat out there on YouTube, Facebook? Drop them in there. Let's get them answered, because I think I need to go to bed soon. I'm tired. I got to wake up early and go to work too.
And if anyone's curious, these are some of the tubs I use. And basically, they're just clear plastic tubs. The filters came from Micropose, which I follow them on Instagram, but they have a very cool webpage, and they pretty much specialize in filters. And they'll either do filters for tubs, or they'll do like lid filters, and they even have like self-heal injection ports. So they're a really cool resource if you're looking to kind of like mod some of your own at-home stuff.
Cool. Do you ever taste a difference in any of your different substrates?
I haven't yet. Although I do know, I've seen several people on Facebook looking for gluten-free mushrooms, and specifically mushrooms that were grown on a gluten-free substrate, because they claim that the gluten can basically be absorbed and passed through the mushroom. And I've seen a lot of people naysay it, but I've seen a lot of people confirm it as well. And I mean, that's something that, in my opinion, I mean, we found that cordycepsin can be passed from chickens into their eggs. We've really found that a lot of like proteins and different chemicals can be absorbed and passed on by different organisms. And I think that a fungus would probably be the most likely candidate to be able to do that, especially since they produce so many metabolites. And just kind of, if they can't digest something, they'll store it and then eventually just kind of sweat it out. So, I mean, I see it as highly, highly probable that that's happening. Whether that would affect the taste, I'm not sure. But I do think probably different nutritional contents would affect the taste. But I personally haven't noticed any.
Someone on the YouTube is asking what the diamond fertilizer was called.
Oh, so it's General Organics is the company that makes it, and it's a part of the Go box, the General Organics box, and it's called Black Diamond, and I believe it's like a 0.1 or something like that. So it doesn't have any nitrogen or phosphorus. I don't think it has any potassium, but it's pretty much just humic acid from peat bogs. So jar or bag when growing mycelium. I started off with jars. I really like jars mainly because of the reuse factor. I am doing a lot of bag cultivation right now, especially just to get familiar with it and to kind of watch things. It's really kind of difficult to make a giant cleaning operation for all the jars. But it is something that I really, I'd like to see more large jar culture, although that does get heavy. Or to create some type of new like tub that's made out of like clay or something like that to cultivate in. But overall, I think that at least like shape and design wise, I think bags are a little bit better because you can move them and you can punch them and you can redistribute the mycelium a lot easier than you can in a jar. So like if there was a perfect world with a green alternative, and like could mushrooms reproduce like replace glass or plastic, I would probably say like the plastic bag design, at least in my opinion, is really, really good for redistributing your mycelium to get it to recolonize faster, and gives you kind of a lot more control on patch size, how much air exchange you want, and kind of really controls where you can put different attachments. Whereas obviously with a jar, you can only use the lid for like an injection site or a patch.
Yeah. I really like, just to kind of echo that, I really like the bags too for a lot of the reasons you articulated. And you can also, if you're like trying to do it on a commercial scale, you can just grow more of that grain spawn than you ever could in a jar in one of those
Mhm.
bags. Like it's like a huge upgrade, and it makes the workflow down the line when you're like inoculating
Yeah.
substrates so much easier than having to like break up a bunch of half-gallon jars. And and also jars take up a lot of space. My whole living room is like filled with half jars that I bought a whole bunch, half-gallon jars. I bought a whole bunch of those, and then now I'm not really cultivating, but I'm not going to get rid of them because I want to keep them around for cultivation. And so now they're just like sitting there collecting dust right now. Whereas like I do have some bags and they're in a box, with
They're
stored with the rest
compressed.
of the
Yeah.
mushroom stuff. It's like it saves so much space. And yeah, I really wish that we can figure out a way to keep that, maybe like silicone bags or something like that, but like a reusable factor, but the malleability and the portability and the just like,
Mhm.
I mean, you can use the bags for spawning and fruiting. Like
Mhm.
they're diverse, and you can get more mushrooms out of. There's more diverse uses for the bags in the mushroom farming context. So you can get more mushrooms fruiting from the bags than you ever could out of jars, just because like you need that mass of substrate for them to grow off of. And so
Mhm.
like I if you can kind of offset that by having a bunch more jars to grow out of, then yeah. But it is the sad reality that like there is a lot of plastic trash in
Mhm.
a lot of the specialty mushroom cultivation that goes on.
Well, and kind of like what I've noticed too, especially with trying to cultivate new and like wild forage mushrooms that people haven't yet, is like really controlling the amount of exposure you have is infinite with a bag. You can cut a hole in it. You can just do a tiny slice. You can do a pinpoint. And so you can have all these different fruiting or evaporation surfaces that you really can't do with a jar. You can't make a jar with tiny openings or large openings or different types of openings because it's all got to kind of be a jar shape.
Yeah, and
Uh, oh, sorry.
oh, I was just gonna say, and like certain things about jars too. Like certain mushrooms, like they fruit weird out of them, so like they don't look good on a farmer's market
Mhm.
table or on a store shelf or something like that too. So, yeah, there's certain constraints to jars. I mean, obviously, the biggest benefit is like their reusability factor, and that increases the sustainability of the mushroom farm up a lot as far as like environmental impact. But it does come with a lot of drawbacks, in my opinion. Also.
And then I was gonna say, someone said, how concerned with measurements when spawning? Um, so honestly, I've been trying to take a lot more measurements because I've noticed that it seems like like almost everybody I know doesn't. And so then and I don't know if it's to protect trade secrets or if it's just kind of like we just kind of eyeball stuff a lot. But I know like most of the people when I ask them, especially through like online interactions, people are like, I just kind of eyeball it. I just kind of have a feel for it now. I've been trying to take measurements of it, especially because I've been introduced to biologic efficiency, which is kind of like how much water is being consumed and how much spawn or substrate is being consumed, and then mainly looking at like how many pounds of mushroom did you get per like grain per ground of dry grain spawn or per grain per amount of like dry wood. And so I've started to get into measurements, but really a lot of it does have a pretty wide tolerance, and just kind of building an eye for like what's too wet and starts to grow bacteria readily, and like what's too dry and dries them out. Most people get that at least currently, just from kind of enough failed experiments. And I think that's because most people just aren't writing everything down when they go into kind of making spawn or experimenting with different spawn. So that's something that I've started doing, especially because with the tea kettle, I know that I'm almost always using like 1.5 liters of water, and then I'm either doing that like once or twice. And so then it's just kind of a matter of like weighing out your dry material beforehand. And so I think um, out of 8 pounds of bird seed, I get about 12 bags, about 12 3-pound bags. So that would be like 36. So it's a significant amount of water that's being added. But again, that's something I'm trying to start writing down, especially with the substrate too, because the substrate is much less and just absorbs water like crazy, as opposed to like a grain that's having it forced into it. But if you are looking for very specific measurements, I know shroomery.org has a lot of posts where they actually go into, I don't know if it's always exact numbers, but they do a lot of volumes. So like the amount of volume per grain versus volume per water. The biggest setback when growing outdoor or indoor, I would say outdoors is definitely competition, and whether that is molds tend to land a lot easier. So anything that's kind of been coddled in a bag really tends to not be ready for the outdoor environment, which sometimes it is, and sometimes it can do pretty well. But from my experience, either another mold or bacteria gets to it, or in this area, some sort of mammal. If it's left outside, there's a lot of mammals that like mushrooms, a lot of foxes and possums that'll come dig into beds to dig it out and eat it. We'll dig into bags. I've had several hardened reishi, almost like sculptures on my porch, and they've dug through the tough leather of the reishi to try and get at the grain spawn inside it. Indoors, I would probably say environmental controls, regulating your like CO2 buildup, getting your humidity and temperatures right, is really kind of the difficult part of indoor growing, at least because you're producing consistently, whereas outdoor is seasonal. You're just kind of waiting for the right conditions to set, as opposed to trying to control those day to day. But I would say those are probably the two biggest setbacks for outdoor and indoor.
So this question is from the YouTube. If you make too much substrate, could you freeze it and the excess and use it later?
Absolutely. And honestly, I know a few people who don't even freeze it. Unicorn Bags is very fond of saying sterile is sterile. So once it's been sterilized in like an all-American pressure cooker, I mean, in theory, there is nothing alive in there, and there will be nothing alive in there until you put something alive in there. That being said, our recommendations for sterility don't really match everything that can be done in nature. Like obviously extremophiles, I mean, 20 or 90 minutes at 15 psi is nothing to something that lives in a sulfur vent at the bottom of the ocean. So there are always things that can exceed that general sterility guideline. But if you do run it for long enough in a pressure cooker, in theory, nothing should be able to contaminate it, at least while it's on the shelf. Obviously, when you open it to add spawn, then that would be your really only time that it could be contaminated. Cost versus profit, outdoor or indoor, that's really going to depend on kind of how much input you want to put into it. So if you can be monitoring it all day and you can be regulating a temperature all day, indoor is definitely the way to go. You can produce a lot of mushrooms in a very similar climate very, very quickly. However, if you don't have a lot of time to manage it and you don't have a lot of material ready for you or to you, outdoors very, very great because it's kind of a set it and forget it mentality. You could go plug a whole bunch of logs, kind of like in that one that was growing a bunch of wood ears. Literally just have a small field set up with logs on logs on logs that have been inoculated, in like a weekend or two of work. And then you pretty much just wait for the right season for all those to flush, and then you go harvest them. So really it's just kind of about how much do you want to put in getting the operation going and running.
Yeah, indoors, especially if you're trying to be like a at scale farm trying to generate revenue and pay bills with that stuff, it's gonna take a relative high amount of like upfront costs. But like Ryan was saying, you can really get pumping mushrooms pretty quickly. So if you can cover that cost, like usually the market's there and people want it, especially if you're like in an urban area and there's restaurants around and stuff like that. And then another thing that was drawing to me into like trying to start a farm, a while back, and didn't work out because I couldn't find the right space for myself at that point. But oh, I forgot where I was going with that. Darn. um, yeah, shoot, maybe it'll come to me. But like I said, it's been a long day. Landscaping. And we're at 9 o'clock now. So if there are no more questions, um, yeah, I think we'll wrap it up.
Cool.
Sound good, everybody? Last chance for questions. I got to give the YouTube a minute because it's a little bit behind
Gotcha.
on the restream. But yeah, cool. Well, thanks everybody for tuning in tonight on this Central Texas Mycological Society's monthly lecture and conversation. And if you like this, obviously like the video, hit the like the little thumbs up there, and if you want to see more of these, hit the subscribe button, all that good stuff. Follow us on Facebook, YouTube. Head over to our website. If you want to support us financially, you can make donations there or become a member and get access to members only Discord for chatting about stuff, members only forays, advance notice on any cultivation classes, identification classes that we'll be doing. Like I said earlier, we'll be doing in-person stuff coming really soon as more people get vaccinated and the COVID numbers keep improving. So yeah, keep sign up for our newsletter, stay in touch with us on Facebook, the Instagram, the Twitter, all those places, because we're trying to be everywhere so we can get as many people interested in fungi as possible here in Texas. Yeah. So thank you for tuning in. Good night, everybody.